Thank you for the introduction, and thank you for the invitation to this wonderful week. Can you hear me well? Yeah? I guess so. So in this presentation, I will try to explain you how we can use high-dimensional statistical inference to improve our understanding of the brain. So I will focus on brain imaging, but I would like to say that this setup exactly would apply to medical imaging in general, if you want to make a prediction from a brain image, so like a diagnosis application, or even in genomics, when you have high-dimensional complex data with some structure, like in genetic data, and you want to predict some outcome for the person. It's a very similar setup. So I will restrict it to cognitive brain imaging, but actually the technique that I will discuss are much wider in terms of application. So cognitive neuroimaging addresses the question of how our brain activities are influenced, affected, or controlled by neural circuits in the brain. So how activity in a certain brain region will affect your cognitive activity, your thoughts. So typically, this kind of question is addressed by an experimental setup in which cognitive neuroscientists come with cognitive theories that they want to probe experimentally. And for that, they present some material, like stimuli that will be presented to participants, auditory or visual, or some tasks that people will have to do. And you organize this condition, this stimuli, in a so-called experimental paradigm. And then you take a fresh brain of a 20-year-old student, you put him or her in a scanner, and you get this brain activity. So the brain responds to the tasks that were presented. I have to say immediately that the data, so here I'm talking about functional magnetic resonance imaging data, fMRI data, and these data are very noisy. You know, in computer vision, you can take an image. Well, typically you can say what's in the image. If you look at an fMRI image, you cannot say exactly what the subject was doing just by looking at the brain activity of the subject, because there's too much noise to make it visually conspicuous. So for the statistical analysis of this type of data, you've got two typical problems. One is encoding. So you try to explain brain activity given the content that was presented. So it's a forward model, if you want, or decoding, which aims at predicting which tasks were performed, which objects were presented, given brain activity. So I will first insist on these two concepts. Encoding has been done for like 25 years now, since we've been using functional magnetic resonance imaging. And typically, we can use batteries of tasks to assess lots of conditions and look at brain activity differences between two conditions. We call them contrasts. And then for each of these contrasts, which I described here with terms, you have a corresponding brain map with colored, which tells you which brain region are involved in the task somehow. This is an average over 80 subjects, so it looks pretty nice overall, but remember that for each unique subject that you would scan, the images would be very noisy. This does not work well. Okay, one other point that I want to put forward before going into the real matters is that resolution increases. So 15 years ago, we were scanning with a resolution of 3 millimeters in the fMRI. Now we are about at 1.5 millimeter, which multiplied by 8 the number of voxels, of special units that you are observing. And in the coming year, we want to arrive to 500 microns about, which would then, so to cover the brain, you would need about 10 million voxels. So, of course, this is important to increase the specificity of the findings, the local specificity of brain structure. But on the other hand, you can guess that for statistical analysis, this is really a burden. Okay, my talk was planned at 11, so to catch your attention, I put a menu, but maybe it's going to fail since we are in the afternoon. So the menu for this presentation will be first a discussion of a causal framework for brain activity decoding. This will be actually the motivation of my talk. And then, so it's really the why question. And then in the next part, I will describe the how part. So how can we make inference on the data? What are the steps that we propose to do? And how can we carry out heavy estimators on this brain imaging data? So let's start with a bit of causal reasoning. When I say causal here, I mean in the sense of structural equation models like models of Judy Pearl. Typically, when you think of the data that we have, so you have some task that was proposed to the subject, which induces some brain activity. So you have a model that can describe brain activity X given the task T. And then, or maybe it's another alternative, you could think as the behavior, what was done by the participant, as a consequence of brain activity. And so you might want to predict behavior from brain activity. It's kind of two sides of the same coin, if you like. But you can actually also analyze the data in an anti-causal manner, trying to decode the behavior that was performed by the subject looking at brain activity, or predicting the task that was presented given brain activity. It's somehow less logical, but as we're going to see, it's interesting quite often to do both directions of inference. So if we formalize a little bit more, let's call T the task that is performed, and our brain will be X, and the brain will be observed in a P location. So P is typically 100,000, if you like. And so we have the signal in all the brain voxels. The model that the way we conceive the data is that the task will have some influence on different brain regions, but also brain regions talk to each other. So they exchange information, and this information exchange is instantaneous with respect to our observations, because fMRI is essentially slow, much slower than brain activity. So what you observe is a network activity that follows a given stimulation. And so it's the result of all these interactions between all the P variables. The statistical analysis then is, for the encoding model, consists in testing for each variable xi whether it's associated or not with the task. So you try to test conditional independence relationships, marginal independence relationships between each brain variable xi and the task. When you do decoding, you want to see if the region xi, the voxel xi, predicts the task, but predicts the task given the activity in all the other voxels. So now you are studying the conditional association between xi and the task given the activity in all other voxels. Same thing if you analyze behavior. Now the direction of causality has changed, but the inference problem is essentially the same. You might want to study the encoding problem, how behavior is associated marginally with each variable, or whether it's associated conditionally on all other variables of the model, of the problem. And now we can start thinking a bit more what this kind of inference will tell us on the link between brain regions and the task that you propose to the subject. So assume for the moment that you are just in a task setting. So the task is here. It may have influence on different brain locations, and also brain locations exchange information. So assume that you do the encoding analysis. So you just study the marginal association between the task and each xi, and there is no association. Then you can simply say that the voxel xi, or the activity in xi, is not an effect of the task. If there is oh, sorry, there's an error here in the two rows. If it's associated, then you can say that the activity in voxel xi is actually an effect of the task. Now, if you were doing a different analysis, still with the same model of the data, but you do a decoding analysis, then you cannot draw the same conclusions. For instance, if you find that the voxel I is independent from the task conditionally on the other variables, then you cannot conclude, because it could be that, well, there is no effect between them, or it can be that the effect is the effect is mediated by another variable. Like, for instance, the task would have an influence on x1, and then x1 would influence x2. So conditional on x1, x2 is not, does not predict the task. And same thing, if you can reject the conditional association, you can still not conclude on the role of the voxel. And you can do the same kind of inference if you analyze behavior. Now, the problem is that here, in many cases, in all the red cases, you cannot really conclude about the direct implications of between the task that was presented and the activity in the voxel. But if you do the two types of inference simultaneously, and you consider the results of both, then the result is much more encouraging. For instance, if you, in the case of a task that was presented, and you look at the response to the task, if you can reject both the marginal and the conditional independence hypothesis, then you can fairly conclude that the activity in region I is an effect of the task, etc. I won't discuss all the cases because it's a bit long, but there remains only one case which is inconclusive, which is the case in which you, in a behavioral experiment, you can reject both null hypotheses. In this case, you cannot conclude because it could be that region I directly has an effect on behavior, so actually creates behavior. But it could also be that there's another region X2 that explains behavior and that explains the activity in voxel I, so that you cannot actually decide. This would support both hypotheses, and you cannot still decide between the two. But what I'm telling you essentially is that when you want to understand the relationship between brain activity and task, you probably want to do both encoding and decoding. And actually, before doing all this kind of causal algebra, if you want, empirically, we had found that this was the right way to do brain mapping. We did this work some time ago, where, for instance, we wanted to study the effect of viewing a place image, like viewing the image of a house. So if you look at the impact on brain activity of viewing such an image, you see something like that, with some relatively wide regions, but some hotspots here. Now, if you do the reverse inference, where you try to predict the occurrence of a house seen by the subject conditional on brain activity, then you have a different picture, but essentially taking the conjunction of these two images, so keeping these two regions, actually outlines the regions that are really sensitive to places in the visual cortex, those parahippocampal regions. And we have used it a lot. This is not super interesting when you study a single experimental condition. It becomes more interesting when you study a bunch of stimuli. So here there are 10 different categories of visual stimuli. I don't give all details, but you see that if you just do the simple forward inference, there is a lot of overlap. It's very cluttered in terms of representation. While if you do the conjunction of the forward and reverse inference, you obtain a much clearer view. So you only retain the regions that are clearly conditionally and marginally associated with the occurrence of the stimulus. And you can do that with visual categories or with speech content, auditory content, etc. And you obtain with this a better mapping of the auditory and language brain regions here. So actually, we have published an atlas of brain regions associated with 20 concepts that occur most frequently in the literature. So with 10 visual categories, so response to faces, places, body parts, tools, etc., letters, numbers, etc. Motor task, feet and hands motor mapping, language and audition, and computation and visual saccades. Okay, these are nice images. Maybe this is not really what is of interest to you, but this is really the motivation for us. We need to have good decoding models, so what I could also reverse inference, on which you can study statistically the conditional association between a given brain voxel and the tasks that was presented. So we'll have to do statistics. And of course, you understand the difficulty of the problem will be that we are conditioning on all the brain, all the other variables. So we are actually conditioning the link between a given brain region and the task on too many variables. This makes it very, very hard. So it's a really, I will frame it as a problem of statistical testing in a high-dimensional regression. Actually, for the remainder of the presentation, we will focus on the following model. So Y is the output, so it can be the behavior or the task. We're no longer thinking of these causal relationships. We just think of, okay, in a multivariate manner, so given brain activity, we want to predict Y. And most importantly, we want to make inference on the weight vector that relates brain activity to the task. So we assume that there is a true such a pattern, W star, and that the observation is also noisy. Usually, when you do machine learning, you just want to find a weight vector W that minimizes the error, so that X times W is close to the ground truth. So you want to estimate W hat such that this is small in some norm. Here, we would like to recover the correct W, so we want actually W to be close to the true one. And this is, of course, a much harder problem. Actually, we will want to have statistical guarantees on the difference. We want to have some confidence that we can say that Wj is 0 or non-zero. By the way, here I'm just using a linear model because the problem is already very hard with a linear model. Maybe in the future we will be using multi-layer networks with nonlinearities. Probably when we have more brain images, this will become very relevant. But for today, we stick to simple linear models. So the first thing that we will have to do to achieve this kind of inference will be to reduce the problem because it's not tractable. Okay? You cannot condition on 100,000 variables. This doesn't make sense. So I will quickly explain how we do that. So we have P features. I said P equal 100,000, and we first have to reduce it to a tractable number, so maybe 1,000. A typical way to do this kind of things in machine learning is to use random projections, which works fairly well. Actually, there is a talk about this topic, I think, at the end of the afternoon. We've done it. It works fairly well, but it does not use the structure that you have in images, in medical images in general and in brain images in particular. So actually, you can do much better by using the image structure, for instance, by leveraging the local redundancy in the images. So we'll rather group the features, group the brain locations, the voxels, in a clustering algorithms. So what is called superpixels in computer vision. And to do that, we need, of course, fast clustering methods that work fast when you want to obtain lots of clusters from your data. So the idea is the following. You've got a bunch of images. So here I take a computer vision example because it's easier to figure out. By clustering, you create some groups of clusters that are neighbors, especially, and that are very correlated. And then you can actually replace each image by a local average in the different clusters. So This is essentially an operator that you apply in the image. So the operator essentially reduces the image to the values that you were observed in the different clusters. But by using the transpose operator, you can actually obtain a piecewise approximation of the input image. It's just a rewording of what people have been doing with superpixels. Now, if you just quickly study the properties of this transformation, obviously, when you replace an image X by a piecewise constant model by doing local averages, you are applying a compressive transformation. So it's obvious to see that the transform of X will be slightly smaller than X in terms of L2 norm. But the good news is that the difference is not huge if the signal is very smooth. So here we introduce an hypothesis of L Lipschitz for the images. So the difference of signal between two pixels or two voxels is bounded by the distance in the image space of these pixels or voxels. So if the two pixels are very close, their signal is likely very similar. So if you've got this type of property, then you can see that our approximation is not too bad. The error can be bounded above by the following, something that involves the Lipschitz constant and the size of the cluster that you used. And actually, you can quickly see that to have a good representation, you need the clusters to have more or less equal size. Actually, the situation can be a bit better than that, because quite often what you observe is a mixture of signal and noise, where the signal is smooth and the noise is not. It's the case with MRI, because you've got some additive observation noise, which is white. So in that case, you can easily show that the mean squared error of the approximation of your signal, so the piecewise approximation, is bounded by the term of two terms, a bias term that is essentially related to the Lipschitz constant, so the bias that you have by replacing the pixel values by the local clusters, plus an error which is the variance term, which is the additional noise. But this term is reduced when you take a local average of the signal, which means that you can even be in a fortunate situation where your approximation is a more accurate representation than the original signal. And this occurs if the Lipschitz constant is small enough with respect to the level of the noise. And actually, we can go a bit further. If we assume that the clusters have equal size, we can find some scaling. And this gives you actually the number of clusters that would be optimal to compress the image in this type of setting. And I'm telling you this because this is the case. This is what we observe in the data. So first, we need a fast clustering method. So people use superpixels in computer vision. We found an alternative that is faster and in a few iterations, in just 3 passes on the image, gives you a lot of clusters which summarize the signal quite well. It's just a recursive neighbor agglomeration method. And when you do that on a database of faces, so the Olivetti faces, so you can do that with a slick algorithm, which is state-of-the-art in computer vision. This is our approach, RINA. It's kind of similar. Maybe slightly more noisy, slightly less regular than the slick, but it's much faster, and overall it's doing good. Now, if you, so what we have checked first, when we did that work a few years ago, we were not interested in inference in the first place. We were just using this reduction to make machine learning more tractable. So if you take a large databases of images and you want to predict some outcome, it takes time because each image is very heavy. So assume like 1,000 images, then it becomes very slow in terms of computation. So just doing a simple L2 logistic regression, so the purple curve would be the vanilla method for logistic regression, Well, on this data set, it takes you about 2 minutes to get the solution. With our approach where you first compress the image and then input it to the logistic regression, it's about 10 times faster. You get the solution in about 20 seconds. On a much larger data set, similarly, you have the vanilla solution that runs in about 30 minutes, and you can get a solution as good in only 2 minutes using this compression ID. Actually, it's even better because the asymptotic solution is actually higher. So the accuracy is higher with our approach because of the positive denoising effect. So this is good. We need to go then one step further, which will be to create this model. So you understand that these models are fast, they are efficient to represent image data. But we will add something upon this. We will ensemble the models. So I recall you our setup where we do the prediction based on brain images. So we predict Y from X. We can use clustering to create contiguous regions. And then we can solve the regression problem to predict Y in the cluster-based representation. For instance, you can do that. Here I use a lasso regression to estimate the weights that predict the occurrence of whether the person is viewing a face or a place image. Okay, I give you this result, but as always with a lasso estimator, you should not trust it too much because lasso tends to not select very well the relevant regions when there is correlation among features, and there is correlation among features. And also, this result depends on the clustering that you've chosen, okay? I've picked one method for clustering, you have used it. Now pick another one, you obtain another solution, or pick another one, you obtain a third solution. So which one is better, we don't know. So probably you don't want to trust any of these resulting images, but what you would trust rather is the average of this solution. Probably the average is closer to the truth, if any, than any of your models. So this is where we introduce an ensembling step or aggregation step in which we essentially average the models. And actually, you can do that with any kind of estimator. Here on a similar problem where we predict the image viewed by the subject, if you use an SVM classifier, you obtain a non-sparse or rather messy brain map. If you do this trick of clustering and solving the SVM-L2 on the clustered representation and then repeating the thing several times and ensembling the solution with different clusterings, you obtain a pattern which is as good as the first one in terms of accuracy, but much easier, much clearer in terms of interpretation. So we call this approach fast regularized ensemble of models, so FREM. It can be quite efficient computationally to run these kind of things because the learner that you are using, which takes you time, like the Lasso CV estimator, which is very expensive due to the nested cross-validation, is run on a much smaller data. You have reduced the data at each iteration. And another trick that helps is just to say when you do the cross-validation, so the internal loop, you will use several hyperparameters for the lambda parameters of the Lasso estimator. Instead of picking the best lambda, you may just want to pick the best model. So for each fold of cross-validation, you will just pick the best linear models that you had among the different lambdas that you probed, and you just average these models. And with this, you don't have any refitting procedure, which makes it more efficient. Actually, it's the idea of the people in Orsay called it the AGU approach. It's a kind of mixing of bagging and cross-validation. When you do this kind of thing, first of all, you don't lose accuracy. Here we have benchmarked extensively all kind of learners to predict what was seen by the subject. So here I just give you prediction accuracy. So each color represents a different data set. So we have benchmarked maybe 20 data sets, and we obtain the score for each cross-validation fold. And so we represent then the box plot of the differences in the result. We see that the learners that rely on the frame trick do at least as well as the other ones. So some of them are very cheap, some of them are more expensive. You don't see much difference actually in accuracy across data sets, but frame tends to improve accuracy. Not hugely, by the way, but in general, it's a positive trend. More importantly, this enhances the weight stability. If you do a half-split experiment in which you run the model on two different parts of the dataset and compare the weight maps, remember that at the end we want the weight maps. So you see that the weight maps are much more similar when you use the frame estimator. So for instance, here in this problem, the non-sparse models are much better than the sparse models. So the non-sparse are in white here. And you see that if you take the SVML2, well, you can actually improve the stability of the map just by using this trick of frame without or with clustering. Or if you take a sparse estimator, the stability is crap, but you improve it by the bagging ID and then even more with the clustering ID. So kind of grouping features randomly essentially improves the stability of the map. In terms of computation cost, we are, of course, a bit more expensive than the initial estimators because we had to do that on several times in the bagging experiment. But we're still less expensive than complex convex estimators that enforce spatial regularization, like minimizing total variation of the weight maps, for instance. So it's a good compromise between accuracy, stability of the results, and computation time. Okay, I'm almost running late. So now I will go to the main part. We know how to estimate good feature weights from brain images for the decoding problems. I need to explain you how we can do statistics on that. How can we control the occurrence of false positives in these maps? So the inference that we are making is try to find the voxels in which we can say with some certainty that the weight is non-zero. This is the inference we want to make. And we want a p-value on that. We want a statistical guarantee. So in the literature, solutions to get p-values on high-dimensional linear models, well, there are not many. So the group of Bullman and Meinshausen have worked a lot on these ideas with the so-called corrected ridge estimator or the Desparsified Lasso, which I'm going to present now because it's the most powerful solution, I think. Also some multi-split ideas or knockoff inference, but they are less powerful, I think. And the common point of all these methods is that they can work for p maybe a bit larger than n, but not too large. When p is much larger than n, they're all going to fail. So the Desparsified Lasso, maybe some of you know that. It's a generalization of ordinary least squares regression when the number of regressors p is larger than the number of samples n. Actually, if you go back to vanilla ordinary least square, you can actually write the solution, the weight vector. So assuming that we are in a well-posed case, we can actually write the solution as zj.y divided by zj.xj, where zj is the residual of the regression of xj on the remaining regressors. So all regressors but xj. Okay? And this is a key to generalize to high dimension. So in high dimension, we just need to find a surrogate for zj that is well defined, and it's very simple. In this projection on the x minus j, you have a regression problem involved. You replace the ordinary least square regression by a lasso regression, in which case it can run with a very large p. And you can then define zj even when p is very large. So doing that, you can obtain an estimator for the weight vector wj hat. you need, so it's a ZJ obtained with a lasso regression for the residual estimation. There is a small correction term which is kind of debiasing due to the fact that we are estimating a WJ in a setting where n is smaller than p. This disappears when n is smaller than is larger than p. Sorry. It requires an initial estimation of W here, which we essentially take as, for instance, as a lasso estimate. The good thing with this estimator WJ hat is that it has a covariance matrix associated. We can at least have an estimate of the covariance of WJ and WK, so I call it omega JK. Actually, we don't need the full matrix, we just need the diagonal terms. And with the diagonal terms, you obtain a scaling that tells you how far your estimate WJ is from the ground truth WJ star. So in this scaling model, you've got the term the omega JJ that comes, and an estimate of the noise, and there are some standard estimators for that. So now you can have confidence intervals on your solution and actually take a problem where n equal p. The ordinary least square solution is no longer well posed, so it completely blows up. So here the true WJ is a red signal, and in blue you have the estimate using ordinary least square. You see that the estimate is very jittered and the confidence intervals are huge, which means that you cannot separate the signal, the non-zero WJ, from the zero WJs. With the Desparsified Lasso, it works perfectly well. Well, at least the confidence interval seems reasonable, and the solution is usable. But still, as I told you, it blows up when p becomes super large. Now take p equal 2000, n equal 100. Again, the Desparsified Lasso cannot deal with this type of setting. So what you need to do then is to introduce some hypotheses to tame the variance somehow. Here this is where we introduce the clustering of the features. So if you take a piecewise constant model with you have less values in your model, so you compress the model, and the clustered Desparsified Lasso works well. As I have told you, we won't be happy with one clustering. We will typically do that with different clusterings and ensemble the solutions. So you obtain a clustering of the images with a method that you prefer. Actually, you use several clusterings. And then you do inference on the clustered representation. So you obtain a WJ hat for each clustered representation, and each voxel will inherit essentially the p-value corresponding to this WJ hat. And then you just need to aggregate the p-values across the clustering. So this is the ensemble of clustered Desparsified Lasso. When you do that on your data, so this is an experiment in which we want to discriminate whether the subject is viewing a face or a place image. So these are different solutions. So it's different solutions of clustered Desparsified Lasso. When I represent the weight maps in a z-scale, so normalized as a Gaussian variable, and you see some hotspots coming, but depending on the clustering chosen for your model, you see that there is a huge jitter in the solution. The huge jitter means that your problem is very ill-posed and that it's very hard to estimate the correct weight maps. So what we do is to aggregate all these solutions into one using the so-called quantile aggregation method of Bullman, and we obtain these weight maps, which seems to be a reasonable consensus of the solutions. Okay, so this is just restating the idea that we are, the path that we are following. Oh, Alex told me that I need to present a theorem in my presentation, which puts me in a hard situation. But actually, so we did one for you, which is to say that the method controls the delta family-wise error rate of the problem. So I need to define, sorry, the delta family-wise error rate. So of course, you cannot, if you look at the errors on the estimation of the support of W, you are going to make mistake because you are accepting a voxel as active when it's often in a cluster that is active. So typically the solution is not estimated in a voxel, it's a local solution. That's why we call it the delta FWER. It means that when you say that a voxel is active in that case, it means that it's in a region where there is activity. So you can only reject the null hypothesis on a dilation of size delta of the true support. This is essentially what this means. And so family-wise error rate is controlled, for the delta family-wise error rate is controlled if for each threshold alpha, we actually have the desired number of false detections, or less than the numbers of less than alpha false detections. And the proposition is that the ensemble of clustered Desparsified Lasso, when it uses clustered with a diameter less than delta, controls the delta family-wise error rate, which kind of makes sense if the hypotheses of Desparsified Lasso are fulfilled. So this is a picture. If the true support are the blue and red regions, so red means positive signal, blue means negative, you cannot say anything about the voxels in white, so those in the vicinity of the true support. But for all the voxels that are far away, you should not detect them more often than what the family-wise error rate tells you. So we have, of course, controlled that on a lot of simulations using cubic brains with 5 regions positively or negatively activated. And we observe that the ensemble of clustered Desparsified Lasso tend to recover quite well the informative regions, much better than the clustered Desparsified Lasso. And we can quantify this with precision recall curves. It's indeed much better. The ensembling always improves the solution and helps controlling the family-wise error rate. And on real imaging datasets, relatively big-ish datasets, we obtain a very reasonable map here predicting some effects of social cognition, like animacy detection in short movies, visual feature discrimination, or obtaining the language network of the brain. Okay, so to summarize, in this presentation, I showed you that causal reasoning about the relationship between cognitive activities and brain activity involves some conditional association analysis. High dimensional, so large P data bring important challenges. One is computation cost, and you should never neglect that. And the second one is the difficulty of statistical inference, what I called conditional association analysis. The solutions to that are ensembling, taking weak learners and ensembling them together. It always improves the solution. Compression, essentially, is a good idea. So we have introduced some efficient stochastic regularizers based on this clustering idea. And we're actually showing currently that this works even in deep multi-layer models and nonlinear models. When you work on images, this kind of regularization with a bank of filters that essentially simplify the images are always beneficial, actually, for convergence and accuracy. Currently, we are comparing this approach with the knockoff ID of Candes, but so far, the knockoff do not perform better than the Desparsified Lasso. We just need to extend it to classification setting because the Desparsified Lasso only works in regression. And also we are comparing it with bootstrap approaches. We provide these tools in open source software, which is very important to make it usable by the neuroimaging community with open source software. So I just list those contributed by my team. And with this, I would like to thank my co-workers, especially Jérôme Alexis Chevalier, Andrés Soyo Sidrobo, and Bin Nguyen, who did most of the work. And this is a collaboration with Joseph Salmon, Sylvain Arlot, and Mathieu Leral. Thank you for your attention.
Okay, so some questions for Bertrand. Okay, just wait for me to go.
Thank you for your interesting talk. At the beginning of your talk, you mentioned that there is a lot of noise in the image. Has this noise been characterized? What is the type of this noise and so on?
Oh, it's complicated noise because you've got acquisition noise just by the MRI device by itself. And then you've got lots of physiological noise because when the subject does the experiment, he or she will press button, he will also brace, etc. And all this induces motion, which is then structured noise in the brain signal. So this results in relatively complex structured components in the images. So the noise is non-local, it has some very complex structure. So it's a mixture of different sources, and it really depends on how well the subject behaves in the scanning session. And then there's another type of noise that comes on top of it, which is the variability between brains when you pull data from different subjects, different studies, different scanners. And so it's another type of noise that comes on top of it.
Just maybe a naive question, but the difficulty is that your Desparsifying Lasso is not an optimization problem. So it's not possible to extend it trivially to other tasks.
Which task you mean?
Like a classification task or like beyond regression?
Here it really relies on the least square loss that is used. I'm not aware of any generalization to the logistic loss, for instance. Okay. But the logistic is quite well behaved in general, so it's possible to think of generalizations. So one of the advantages of the knockoff framework by Candace, so it's not very different in terms of results. The principles are completely different, but the results are not so much different. But the knockoff IDs by Candace work for any type of loss. So you can use whatever, as long as you can say which features are important for your problem, that's fine.
But in practice, it doesn't perform as well, is what you say? Or?
Yeah, I think okay, we're still experimenting on that, but overall, I think that the Desparsified Lasso is still the best solution. Yeah.
Okay, so wait for me.
Thank you for a very interesting talk. I was wondering if, as an alternative to the Desparsified Lasso, whether it's possible to use, another way of removing the bias from the lasso, which is to actually keep the variables that have been selected by the lasso and then perform ordinary least square on the result. Right. So I mean, this is a very
post-selection inference.
Yes, post-selection inference.
So we've tried with the code provided by Stanford, which completely blows up when P becomes large.
Which code? GLMnet?
Just refit linearly.
Yeah, I'm just suggesting.
So they have their approach where they based on estimated cumulative of Gaussians on polyhedra defined in the feature space.
Yeah.
Well, and computationally, this doesn't scale when P is large.
Yeah, so I mean, this is one way, which is relatively sophisticated, but there is an even simpler way, which is to, just refit a simple ordinary least square regression on your on the variables that have been selected. And if you want to make this very rigorous, then you can split the data and have fresh data.
There's a multi-split data, but then still it becomes suboptimal because the multi-split is super expensive. So the multi-split method was introduced by Meinshausen and Bühlmann in 2009. And but we've tried it, but it's not very powerful because you need to split your dataset into one part to select the variables, in another one to get p-values, and overall this is not very powerful. The Desparsified Lasso is typically much more powerful.
Okay, thanks.
Okay, so let's thank Bertrand for his talk.